Our speaker today is Dr. Chris Chase. Dr. Chase is a professor of veterinary and biomedical science at South Dakota State University in Brookings, South Dakota. He received his DVM from Iowa State University and MS and PhD from the University of Wisconsin-Madison, and has been at South Dakota State since 1992. Dr. Chase is the past president of the American College of Veterinary Microbiology and the Conference of Research Workers in Animal Disease. Dr. Chase co-founded RTI, which is Research Technology Innovation, LLC, an animal health contract research organization founded in 1994 that does studies in poultry, swine, and cattle, and is a chief scientific officer and has served as president since 1998. With that, I will let Dr. Chase continue with his presentation.
All right, well, thank you very much, Steve, for that introduction, and welcome this afternoon to this webinar. We're going to talk about prescription platform vaccines, a very cool and interesting tool that we can use for managing emerging and diverse pathogens. So just quickly, the overview of what we're going to talk about today. We'll begin with a short presentation just talking about vaccines, but particularly the immune response that we're looking for with the vaccine. And then we will compare and contrast different vaccine types. And then we'll particularly hone in on the interesting aspects of using prescription vaccines, why we'd want to use a prescription vaccine, what disease best fits those, and then how do you go about getting a prescription vaccine. So let's begin with vaccines and immunology. So there are many different kinds of vaccine platforms out there. We're going to talk about a number of those today, but I think in general, we're used to the killed vaccines, the toxoids. We'll talk about subunits today, and certainly the attenuated vaccines, so the live vaccines. And then at the bottom of the list here, you'll see RNA and DNA vaccines. And certainly we're familiar with those in regards to the mRNA vaccine that's been used with COVID-19. So let's begin then talking about the vaccine response. So with the vaccine response, we're really after two components in terms of what we call the effector functions. So one is sort of, as you see on the left here, the idea that with the antibodies sort of being a guard and helping keep things out. And then on the other side of the coin is the cellular immune response, where we look at destroying the cells or the pathogens that are associated with it. And another way to look at that is in this particular slide. So again, when we talk about keeping things out, typically it can do that by preventing them from even attaching to the cell, or if they do attach to the cell, to prevent them from entering into the cell or being able to become uncoated. And that's really what we often talk about, the term virus neutralization or pathogen neutralization. We're talking about antibody. And that's in contrast into cellular immunity, where we're talking about basically destroying infected cells. And we do that typically by putting holes in them and then just literally blowing them up and resulting then in those cells no longer being able to house the pathogen anymore. I like to think of these vaccines also in a little bit different way, because one of the things I hope that we'll bring home to the message today is in terms of the concept of the antigenic mass. So I divided them up here really into a couple groups. So we've got what I call the fixed antigen mass vaccines. So those are our killed vaccines, the toxoids, the subunits. Then we have the live vaccines that require that the agent replicate. So it has to replicate to generate enough antigenic mass to give us a good response. And you can see there's a number of vaccines that fall into that category. And then a third category would be looking at just nucleic acid replication. And that's what we see, for example, again, with the mRNA vaccines that we've seen with COVID-19. So we're not giving them a pathogen, we're just giving them the RNA, and then the RNA has to be able to produce a protein. So it's actually the protein, the nucleic acid isn't actually replicating, but it's actually producing the protein in the host. So with this little animation, what I want to show you then is just the general idea that we're looking at immune response. So first of all, the immune response occurs in the draining lymph node. So what you're going to see here initially is that we don't have any antigen in our system. And so the brown cells that you see there are T cells and the blue cells are B cells. They're looking around for their antigen, no antigen's present. They're going to head right on out. Now what we're going to see is we're going to see our vaccine come into the picture. So you'll see antigen comes into the picture. And you can see that antigen actually gets into the lymph node very quickly, but a key component is the activation of these antigen presenting cells. And that's where adjuvants and the growth of the virus comes in. So it activates these cells. So as you see them begin to interact with the T cell, now we're getting that vaccine response that we're after. But again, it takes two things here. It takes the antigen and that activation to occur. Oops, excuse me. Anyway, let me just finish that up again. So what you'll see then is, so again, initially no antigen. Okay, so there's no antigen that we're going to see in this particular point. And then we'll see the vaccine come back in again, and we'll again watch what happens as it makes its way through the lymph node. So here comes the vaccine again. And you see the antigen gets to the lymph node very quickly, but again, only that cell that you see that's red out in the periphery is actually going to be delivering the necessary signals. And again, it's going to have antigen on the surface, and it's going to have the what we call the danger signals to activate those B cells and T cells. So here you see them activating T cells. T cells begin to divide. We have enough T cells there, and they come over then to activate the B cell. And that's what you see occurring on the right-hand part of the panel. And so when that occurs, then we have that immune response that we're after from a vaccine. And again, now you'll see the cells move out into the periphery. We'll have memory cells, and we're after the response then, an antibody that we're after in terms of our overall vaccine response. So when we talk about antigenic mass, then modified live vaccines require that the delivery agent, which typically again is some kind of a modified live virus, it will go ahead and enter into the animal, and then it will use the animal as the factory to replicate the virus. And that's what you see in this particular slide, is the idea that we start out with a small amount of virus, it infects the animal, and now we generate antigenic mass. Now, if we look at this in terms of what would happen in a naive animal, again, we inject the virus, it's now going to grow, we generate antigenic mass. The virus is then going to be able to circulate to that lymph node that we just saw in the previous movie, hit the lymph node, and then we're going to develop our immune response. Okay, and and the key here is how much antigenic mass I've got to be able to do that. Now, if I've got an animal that's already had an immune response, okay, so it's been previously vaccinated, and I've already got T cells and antibody that they're there, and you can see that over on the right-hand side, right underneath the skin. I inject the vaccine. There's my vaccine virus from my modified live vaccine. Now, the problem is that immune system doesn't know the difference between a vaccine virus and a wild-type virus. So the immune system sees it, and what it does then is it sort of neutralizes that virus, so we just don't generate a lot of antigenic mass, so we don't get a lot of virus then going over to the lymph node. Now, in contrast with that, inactivated vaccines, and these are things that I'm going to call fixed antigen vaccines, we have to give all the antigen in the initial dose of the vaccine, and that's what we're seeing here. So we give that antigen, it's all in that vaccine that we're delivering. And so, we deliver that again in a naive animal. So now we have lots of virus because, again, it's already been determined the amount of antigen that we're going to have. That then circulates over to the lymph nodes, and now we develop a good immune response. Now, if we look at an animal then that's got active immunity, and now again we're giving more antigenic mass, the thing that we have here is that we've got enough antigen there already. And again, typically with the adjuvants that we're using, that allows that antigen then to get to that lymph node and again to induce and boost that immune response. So again, antigenic mass is the key. All right, so let me demonstrate this with this study that was done a few years back and published by Paul Walls and his group from Auburn. And I'm just going to point out to you a few key components. So we're going to just look at, although this vaccine looked at reproduction as well, I'm just going to talk about what it meant in terms of antibody response, and particularly colostrum antibody to calves. So in this particular study, 2 doses of modified live vaccine were given to all the animals. And then a 3rd dose of vaccine was given where they either got a modified live vaccine or a kill vaccine. So all the animals got 2 doses of modified live, and then they either got modified live or a kill vaccine. And then we're going to look at what the antibody response was to BVD type 1. And so what you can see then with each of the arrows then is where we actually have the original vaccine given. So the modified live vaccine given at the first 2 arrows that are on the left. And then at the 3rd arrow, what we see then is we actually see then when it was either given the modified live vaccine or the inactivated vaccine. And so here you can see that with the modified live, there wasn't much of a boost at all in terms of antibody. And here you can see when they gave the third dose was an activated vaccine, that they had a strong response. But the interesting thing is, when they calved these animals out, and these were beef calves, so they then measured the antibody from the calves, and what you can see is that the calves that nursed the cows that got that one dose, so they got 2 doses of modified to begin with, followed by a single dose of inactivated vaccine, had about 5 times as much antibody as the animals that only got the modified live vaccine. So again, showing you that with antigenic mass, we can generate a stronger antibody response. In this case, we're looking at colostral antibody. So antigen is one key component to any vaccine. The other thing is adjuvant. Okay, so adjuvant is an agent that we add to a vaccine to enhance that immune response. We've known about this for many years, and we know that the interesting thing is that all that's happened, and the good news for us is that over time is that the ability of the adjuvants to do what they need to do has just improved as the technology has improved, and we can take molecules before that maybe weren't particularly immunogenic, and because of the type of adjuvants that we can give, make them be very responsive in terms of immune response. So we know that adjuvants will help increase the antibody response. They'll help induce cell-mediated immunity. They decrease the dose of antigen that we may need in the total amount of vaccine, decrease the number of doses that we have to give to get a good immune response, increase the duration of immunity, and overcome competition between antigens and combination vaccines, again, because we've got an enhancement in terms of that responsiveness. And again, the other thing is in naive populations, hopefully give us a strong immune response as well. Now, there's really two, when we look at adjuvants, we think about this sort of in there's 2 parts to it. One is the carrier, which allows the vaccine to be present, to get into dendritic cells, to be sort of presented over time. And then there's what we call the immune potentiator, which is the second part of that we need. So I mentioned to you before, we need to have antigen, but then we also need to have a signal that tells the immune system really how to kick into gear. And that's really what immune potentiators do. And they do that through something that we call a danger signal. So again, if you think back to that little movie I just showed you, remember there's that cell that came down up from the vaccination site, which was activated, and that was the cell that was presenting antigen that actually turned on the immune response. So we know that in the world, there's not a perfect adjuvant, but if we had one, we'd want one that enhanced the amount of antigen so we could have it over time. So again, to help stimulate that response over time, and at the same time, activating cells. Again, so what we're after is trying to only turn on that part of the immune system that's required for protection and avoid triggering the other unnecessary things that you can sometimes trigger when you turn on an immune response. So in other words, we want our adjuvants to be both effective and safe. Now, as we look at getting a vaccine response, and what I'm showing you here with these little white cells, these are all, imagine, naive T cells. And let's say we're going to vaccinate for BVD. That one blue cell that we see sort of in the middle is that one naive cell that's responsive. And so it finds this antigen, and we can see that down below in terms of the dendritic cell right here. It finds this antigen. That then gets the signal, because it gets that right signal in terms of activation. And now we have an immune response. And so now we've generated parent cells. But what we know is if we have the right adjuvant and the right amount of antigen there, we can enhance that. And that's what I'm showing here. So again, the amount of the T cells is the same. We still only have one T cell that recognizes our antigen, but now we have these dendritic cells and antigen presenting cells. We have more antigen present. And then because we're using adjuvants, we have these cells that are more active. So now, again, we have the same number of T cells coming out, but because we have more opportunity to activate them, we can actually enhance our response and give us a much stronger response. It's going to give us more duration of immunity. It's going to allow us to overcome some of the issues in terms of active immunity, because, again, we have more antigen present and it's activated with the adjuvant that we're using. All right, so now let's talk about the types of vaccines that are out there, and particularly as it relates to some of the new technologies that are available to us. So first thing that we need to realize is that most of the vaccines that we're used to using, that are produced on commercial, are what we call full licensure. So that means that it has to be produced in a licensed establishment. So USDA comes in and inspects it. They have to go through and what's called to develop a master seed. So there's a very firm protocol that's put together in terms of manufacturing process. And then in terms of regulatory approval, they have to be pure, potent, safe, and efficacious. So all those things have to be established. And that takes, again, anywhere from 2 to 6 years and costs certainly more than $1 million to put that together. So again, the key components then on a full license are purity, potency, safety, and efficacy. Now, the interesting thing that we have now, so again, we just talked about this full licensure. Okay, and again, so with full licensure, so all those things that I just talked about in terms of the manufacturing outline, those things are put together, and then we end up getting a vaccine that's approved. And that's what we call fully licensed. Now what we can do, though, is that we can actually take, particularly when we use what we call platform, so we have techniques and opportunities to use technology where we can express these genes in the same, what we call platform, and we do that in a production platform license. What we can do with those is take our gene of interest, and so here we see that there's a gene of interest, and in this case, I'm showing you here's coronavirus, here's rotavirus. So again, so we take, let's say it's coronavirus, we put that coronavirus into our platform, put our gene of interest. And then what we can do with these platforms is that we can go back again, and if there's another gene that we want to put in, swap out that gene of interest and go ahead and make and express that gene as well in the vaccine. But because we already have approved the platform with the full licensure, we can just basically insert them and because the licensing process has already been done. Now we can take that one step further with this prescription platform, though, where we have a valid veterinary client-patient relationship, and we can actually use, again, that same technology, and we can swap out our gene of interest, but in this case, we can actually have different pathogens as well. Okay, so it allows us more utility in terms of what we can actually do. But again, it's because we began with a full licensure, and now we've ended up being able to take this out to what we call these prescription platform licenses. All right, so a little bit more about that. So again, it has to be a non-replicating vector. You do the master seed. So just what we've talked about for full licensure, the whole outline of production has to be laid out. There's already specific studies that are done with the target antigen to be able to, again, show that what the dose is, the route, age, and the species, and all the additional things that you have to do for full licensure. And so after you have that licensure, though, then you can take the gene of interest and you can be swapped out again with the same pathogen, because this is the production platform, and it must be field relevant to that gene of interest. And the beauty of this is there's no changes in the manufacturing process, okay, because you've already established that. So you can just put it in and do what we call plug and play per pathogen. And probably, again, if you think of, again, an example that we have right now, COVID-19 and messenger RNA vaccines fit that bill. So again, you take that, in this case, the gene of interest, you go ahead and inject it. The messenger RNA produces a protein. The protein then is antigenic, and we have our vaccine response. And again, they can come back and take, a new COVID-19 protein and do the same thing and move onward. So that, again, that's the beauty of these platform vaccines is that you can just take that, a different strain and plug these into the vaccine. Now, again, so what's different with prescriptions, though, again, is first of all, we're going to use that same platform. Remember, it's fully licensed. The difference is now we need it because we're doing this for our client. We have to have a veterinary client-patient relationship, and then because we know what the production process is already, because we use the platform, we've already got. We know how the vector and all those things work in terms of the dose and the amount of antigen, and those things are already based on the prescription platform. Or on the original platform, the production platform, and then we can use our gene of interest from different pathogens. So down here you see the beauty is that you've been able to use it. My little example here is you could have BVD, IBR, PI3, BRSV, and BC and bovine coronavirus in there as well. It is restricted, though, in terms of we can't use it with any organisms that are part of a surveillance program or foreign animal disease, because again, we're going to use things that are endemic and things that are actually in the U.S. So again, the big advantage that we have then with these prescription vaccines is again, we have a consistent manufacturing process that's already been outlined and approved. We can use the biologically relevant and timely antigens that we need to use. improve the potential for biological diversity, because again, we can add antigens as we see fit as disease comes out. With new and emerging disease options, there's certainly, that's another advantage that these platforms have. And again, because we're going to have a veterinary client-patient relationship that can be very customer or market specific. And the other thing about it is because we're only looking at a piece of a gene, or some of these viruses and some of these other organisms are difficult to grow. All we need to do is to determine that piece of the genome that we're interested in and express that. Now, the other thing about these is as you compare them to a whole cell or a whole organism versus a subunit prescription vaccine, is that when we're using the whole killed organism or a whole organism, the immune system is directed against all of the proteins. Okay, well, we know that typically there's only a few of the proteins that are really important in terms of protection and giving us good immunity. And so the beauty then with prescription vaccines is that they're only going to target the needed parts for a vaccine. And that in this particular case, we're showing just the hemagglutinin, which again, this is with influenza, and we know that that's the major protein that we're interested in. Again, so we can just really have our immune response pointed at that protein that we know is really important in this case for attachment and getting into the cell of the animal. Now, let's just do a couple comparisons here. So if we look at commercial versus prescription vaccines, so again, we know that commercial vaccines is derived from a whole virus or a subunit. We know they've had a high success rate, but it may be that the targets become outdated as new strains emerge. And this is certainly something that we see with things like influenza, with rotavirus. And again, vaccines can be created as modified live or killed and typically used with an adjuvant. We know that they're mass produced and distributed, again, because they've been in terms of the licensing. But because of the full licensure, there's a longer development cycle and the efficacy studies that are needed. And again, so all those things have to occur. So the prescription, again, the advantage that we have, it's again, we're using the gene of interest. And again, if we have viruses that are hard to grow, all we need to do again is get the gene that we're know is protective and be able to express that. We're using again subunit proteins. So again, it's very targeted in terms of the vaccine that we're using. And again, we're using an adjuvant. And again, we're using a veterinary client-patient relationship for that premises or that operation, so that we're again using something that's specific. And again, we're only using the gene of interest. And because we've already got this platform already licensed, there's a much shortened development cycle, because again, we can do that sort of that plug and play. And the safety is proven. The efficacy, though, is not. Okay, because remember, we're putting a new gene in where the initial product had to be fully licensed with the proven efficacy for the platform. Again, with this, where the safety is going to be proven, but again, efficacy is going to be dependent after we use it on the premises. All right, so now let's compare that then to our conventional sort of autogenous vaccines, where basically there's two of those four pillars that they have to satisfy, and that's purity and safety, not potency and certainly not efficacy. So if we look at comparing then the prescription vaccine to autogenous, so again, we've got the gene of interest in the prescription vaccine, where with the autogenous, we're going to grow we have to grow the whole organism and then inactivate that particular organism. And the subunit, again, they're targeted. And again, with the hard to grow viruses, we definitely have an advantage with prescription vaccines versus autogenous. Again, using in both cases, using the veterinary client-patient relationship, again, targeting it to a specific rather than the whole virus, or whole bacteria. And again, the short development cycles. And then the big thing is that, again, because we're using the gene of interest, there is no sort of master seeds that expire, that like we see with the autogenous vaccine. And again, in both cases, the efficacy is unproven. But again, we have a platform here that we know how that performs. All right. So again, just a little bit more about prescription vaccines then. So where they give us, so a lot of utility is in organisms have a lot of diversity. So yeah, there may be commercial vaccines that are out there, but in terms of, because of the diversity that's within the organisms, there are holes in terms of what we might see. So it could be coronavirus, it could be rotavirus. If you're looking at poultry or swine, influenza A, and then certainly BVD in cattle as well. And then as we know, in different parts of the world, the epidemiology and the strains can be different. So what we're seeing in the US versus the European Union could be quite different. So again, we can make it, more site specific. And again, then we have the new and faster emerging diseases where there aren't any vaccines and they require a faster response. So one of the viruses that we've been dealing with, particularly, in cattle, that's certainly new. And again, we're trying to figure out how that fits in, is an influenza D virus. And then we have bovine respiratory coronavirus, which you certainly have seen issues with that as well. And again, we mentioned this idea about the organisms that don't grow very well, because again, we're using just the gene of interest. We can take that target and again, plug and play it in that platform so that we can get a good response and be able, without having to worry about trying to culture the organism. So again, as this policy, or in terms of this production platform and prescription platform, was a directive that came from USDA. And again, and they've taken this very proactive response to allow new and different registration processes, which then allow veterinarians and biological companies then to utilize these new and different vaccine options. So again, we've already talked, about a number of these advantages, but again, making them, pathogen and isolate specific. So again, where there's a lot of diversity, or there isn't that pathogen, there isn't a vaccine for that, because you've already got, a fully licensed vaccine. It's the platform. It streamlines the approval process, and again, it's focused on just that gene of interest. And again, we're using, a non-replicating, non-viable vaccines in terms of the ultimate response that we're after. All right, so again, the fact that we know how to make a vaccine is not the problem. In other words, people have spent a lot of time, and I've spent a lot of time in doing research to figure out, what's the important genes in terms of making an efficacious vaccine. So we know we've been able to, publish papers about that, do a lot of work in mice or other places, and using other platforms. But the problem is a lot of those maybe not come to fruition, because again, it's things that we do in the research lab, but in terms of how that translates to the market, it can be different. So we know making sort of new vaccines with new approaches. Again, we've mentioned the length of time that can take and the expenses involved in that. And again, the other thing is because it takes some time. So by the time that the vaccine is licensed, has the target changed enough so that the vaccine may not be that relevant to everybody that's in the industry. And again, the other thing the target may have, for example, if it's a foreign animal disease. I mean, there's been a lot of work done on foreign animal diseases like foot and mouth disease, and fortunately, we haven't had to worry about that in the U.S. But again, that's something that that we need to be aware of. And again, this platform technology, again, is an approach that we've seen basically from One Health. So again, the PED epidemic that we had, you can see from the time that the vaccine was developed to the time that the vaccine was licensed, resulted in it was over a year in terms of that. And so being able to take the platform technology. So again, you can if you think about it from COVID-19 or other things, where One Health, things that we've adapted from animals or man can go in both directions again, and platforms have that advantage. So again, why prescription platform vaccines? Again, so they provide options again for new and emerging diseases. So like bovine coronavirus or influenza D virus. Then you have viruses that you can't grow very well, like rotavirus B and C, where those are difficult to grow in cell culture to get up to. Again, remember, antigenic mass is a key. So if I can't grow that virus very well, it's going to be difficult to get the antigenic mass that I need. And again, we're using fully licensed manufacturing processes that have already been tested and been approved. And again, the other thing is because we're using the idea of plug and play, these vaccines can be generated just in a matter of weeks. Okay, and so how do we know what to target? And again, this gets back to our understanding, of the pathogenesis of the organism. So we know that if it's a protein that is a neutralizing antibody that's important in terms of for the virus to get in the cell. We know that, for example, with COVID-19, that the spike protein is important for attachment and getting into the cell. We know for BRSV that it's the fusion in the G protein. So again, we can just work with those. Again, we have published research that points to these, these important targets, and particularly like the example here, BVD, that we know the E2 protein is really the most important protein in terms of being able to neutralize and to protect that animal. And then you've got newer projects or newer viruses where bovine rhinovirus is a virus that we've been doing a lot of surveillance, and we find this virus, and obviously this is not a virus that's in any vaccines, but being able to see again if this is a target that we need to be aware of. Again, this allows us then to look at the proteins, and again, we know what the proteins are important for this coronaviruses. But again, then making vaccines to see if that enhances the response, again, by being able to use the plug and play concept. So again, what we're looking at is with the gene of interest, you've already got a vector system that's already been approved and been the outline of production, all those things are already put together. You then insert that into it. You've got your cell culture system that's already been again approved and fully licensed, because again you're just putting that gene in. Then you produce your viral proteins again. So now we're after that antigenic mass that we talked about right at the beginning of this presentation today. So there are certainly other platform and prescription vaccine licenses out there in terms of Hubie Pharma has a DNA vaccine. MedGenes uses baculovirus. Boehringer Ingelheim has a baculovirus, and Merck has an alpha virus. And right now, the one that's commercially active in cattle has been the platform that's been developed by MedGene. The other thing that's important to understand is that as these agents are detected and they're sequenced, it allows us to generate what we call the master seed bank. And that means in terms of being able to look at those sequences and from a number of different isolates and be able to put those together in terms of, what are the predominant things that we're seeing in the field, particularly as it comes over time, and having those then allows then to actually take those well-known genes that have now been characterized again by sequencing and actually make constructs in that can have those genes in there. And again, it takes a relatively short time from that construct bank to the vaccine, and typically 12 weeks or less in the prescription vaccine to be able to do that. So again, that's a big advantage that you have when you look at prescription vaccines, because we're continually understanding and exploring these different isolates that are out there, understanding kind of the genes of importance and the ones that we're seeing out there allows us to construct these banks to get that again and again, and then looking at what's on the premises allow us to figure out the best constructs then to go with, and then to make our production of that particular vaccine. All right, so this is down to the last slide, and that again, so how do you get a prescription vaccine? So again, the veterinarian submits the sample to the veterinary diagnostic lab, and get in this case so it associate with MedGene with the case, and then MedGene would identifies the genes that they're after in terms of PCR for their virus of interest. Then they go ahead and sequence that gene of interest, and then from the D lab does it, and they send that to MedGene for comparison, and then for vaccine production. For again, the idea of plug and play, that you can take that gene and go ahead and insert it again in that already licensed vector and production system, and then be able to get that vaccine out relatively quickly. And with that, I want to thank you for your attention and ask you if there are any questions. All right, so the question is, you listed BVD in the bank. Is it cross-compatible with BVD type 1 and type 2? And what I would say with that, typically, when you're looking at BVD, and particularly as you look at the main targets, typically you've got to be, because there's enough variability, the type 1 sequences are going to be, it's not going to be in terms of cross-reactive. So you're going to probably want to make sure that it's the type that you're using. What you could do, though, is certainly you could add, you could because you're with a platform, you could plug and play both a type 1 and a type 2 gene as well, in terms of what else that you might be looking for in terms of cross protection, or that might be available. But typically, there's enough difference between type 1 and type 2 that I'd want to make sure that I use that specific type and not look at it in terms of, there certainly are some cross reactivity, but I think in general, in this application, I'd want to be very specific about what I was going to make the vaccine for. All right, the next question is, are there specific BDLs that are working with a company to produce these types of vaccines? And what I would say to you, and I can speak at least for the SDSU lab, I mean, typically, because I think most D labs get requests in terms of isolates, and that's really what this is in terms of being able to, there's an isolate, in this case, it's a matter of the sequence, because again, these might be pathogens that you can't grow. And so then it's a matter of typically, contacting the D lab, and then the D lab, interacting with the firm. But I know that at least at SCSU, we work with all the firms in regards to that, because again, it's the veterinarian that has that veterinary patient relationship who is trying to bring that particular pathogen or that trying to develop a vaccine. So again, I think in most cases, most D labs would work with the company in terms of that, because again, they have a material transfer and those kind of things. But again, there isn't like I said, I know at least in our case, we work with whoever the veterinarian requests. All right, the next question is how long can a farm site use a specific prescription vaccine? Do they need to resequence in 18 months? My understanding, and correct me if I'm wrong, is that with these, because typically what's done, at least in most of my experience, is that often you're looking at a farm and they're doing some fairly continual checks on them in terms of sequencing and that and looking at that. So my guess, I honestly, in terms of do you need to resequence in 18 months, I don't know that with 100% certainty. So I apologize for that, and hopefully we can get that answer out to you, but I don't know that right off the top of my head. So if someone in the audience does, that would be good, and add it to the chat. So the next question is, I missed the introduction. So viruses only or bacterial pathogens? So the answer is, when it comes to being able to plug and play the antigens, you can use either bacteria or viruses. I think the big advantage, at least on the virus side, of those viruses, particularly that you can't grow very well. But again, the gene of interest can be expressed from whatever type of pathogen that you need to do. So that we've been using viruses a lot as the example, and that really probably relates to the fact that some of them are difficult to grow, but yeah, the gene of interest is the gene of interest. So whether it's from bacteria or from a virus, they would both work. All right, the next question is, are the limitations on how big the gene of interest can be for the vector to express? And that's a really good question. And so it gets back to the type of vectors that you're using. There's some limitations with some of particularly of the viral vectors. They're more limited with baculovirus. Some of of some of the other vectors that take bigger gene inserts, and that's not nearly as big a deal. So it really depends on the type of technology that you're using. But yeah, there are some types of the platforms where size could be a bit of a limitation in terms of that. Okay. And then I think there's an answer then to, at least from an autogenous standpoint, if you read in the chat where it says autogenous strains can now be used for 5 years. The 18-month limitation has been ended, but I don't know about the length of time for use of gene sequences. So that's where I'm at right now too. I don't know that definitively. So thank you for that comment. All right, the next question is, how well do the gene expression systems mimic in vivo protein folding? And that's a good question. And in terms of what we know in terms of glycosylation, so one of the things that you didn't see that's used particularly as an expression system, typically with any of the commercial systems is E. coli, because E. coli as a prokaryotic expressing proteins certainly has some limitations, particularly as it relates to glycosylation. Again, depending on the vector, you're going to see some limitations with that. Again, if you have a system where it's a replicating vector and using the cells, the animal, you probably see less of an effect on that. But I would say typically even with baculovirus, which has had some limitations, that people have been able to overcome that by some of the different engineering that they have done as well. And then the next question is, what can you tell us about repeat use of the same vector for different genes in the same animal? Does the immune system start to clear the vector without responding to the new gene sequence? And that would be, again, for those systems that require a replicating vector. Okay, that would be an issue. If you're just giving them a subunit vaccine where you're giving them the protein, then where the antigenic mass is already in the vaccine at that level, that's really not going to have any effect on. But certainly, that's been, at least experimentally, probably where that's been done the most is looking at adenovirus vectors, where has been a concern at times. I don't know that we don't have. I shouldn't say that we have now one obviously for COVID, but I don't know that if anybody has shown in vivo in people that effect. But certainly that's always been a concern, especially because it's replicating. It's sort of the same limitation that we have when we talk about just modified live, because again, if the virus has to replicate or the vector has to replicate, and they got pre-existing immunity, that's an issue. All right, the next question is, can this technology only be used for pre-approved pathogen, or is any virus, bacteria fair game for vaccine development? And so the answer to that is yes, that it could be, for example, rhinovirus, which we isolate fairly frequently, but which there certainly isn't a rhinovirus vaccine in calves. You could certainly, do that again, because you have, the veterinary client-patient relationship in terms of looking at it and in terms of being able to do that. So yeah, it doesn't require something that's already been licensed to do that. And then the last question that's here is, any luck with the different strains of Salmonella Dublin? And I would say that that is a really good question, of which I think that the question was asked earlier about bacterial versus viral, and I would say to you that there certainly are some bacterial antigens that one has been able to use pretty well, but I would say that the gram negatives, particularly Salmonella, I think we're still not to that point with it. So I don't know of any good efficacious vaccines in terms of with the difference, because of the difference in Salmonella that way, in terms of being able to use different strains in these types of vaccines, in terms of the platform. Again, because just these antigens, and the endotoxin and all that, there's a lot more complexity to Salmonella than there is to other pathogens. So I don't think we have I don't know that we probably had luck with that yet. And then the next question, there are no limitations for how long a sequence can be used. So this is back to I appreciate that comment. There's no limitations for how long a sequence can be used. However, it's recommended that the herds to be monitored to determine if new sequences or strains are circulating, at which time a new sequence would be recommended to be added to the product. So again, that's that kind of that sort of that constant surveillance just to see what's going on. And I appreciate all these questions. These are great questions. So thank you for the thought pattern on these questions. Thanks a lot.